Okay. So we are done with explaining about the different kinds of proteins that we have selected, right? And next, so you by just knowing the name of the protein, to some extent you will be able to understand protease. What do you understand by the word protease? It destroys proteins, right? Next, some name of the protein is surface antigen-like protein. The name of the protein itself is telling it is somewhat antigenic. Subtle lysins, peptidases. So you're very clearly you are able to understand by name of the protein itself that little harmful proteins. They degrade proteins like that. And chlamydia, polymorphic membrane protein like protein. Chlamydia is a pathogen. Chlamydia, you have bacteria, right? In bacteria, you have chlamydia. Pathogens they are. So in that pathogen, there is a protein called as polymorphic membrane protein. This is something like that protein. And this was found to be antigenic in some other organisms. And this is one hypothetical protein. We don't know much about this. Then circumsporozoid protein. So by looking at the name of the protein, you don't understand much, right? So you have to do something what to some extent you will understand. To some extent, here, suppose this hypothetical, you don't get any background about this protein by looking at the name. So you have a site called as UniProt. So UniProt is a freely available site, most widely used for protein analysis. In-depth understanding regarding your proteins, right? So you can use UniProt KB site and you just give your accession number to your UniProt. So this is your accession number, right? This is your accession number and give the accession number to the UniProt. So when you give the accession number, just a second. So when you give the accession number, what this UniProt site will do is it will try to give the annotation of your protein. Okay. So what is annotation? I told you. Annotation is little in-depth understanding regarding anything. In general terms also, we use something called as annotation. Annotation is finding out description. Okay. So just I gave you a list of proteins. You don't know much what those proteins might be doing in that particular organism. So it is important that you should feel they might be really useful for our work. So how you will get that information? Right? Actually, if you read one paper, you will get, but you will not have that much time, right? So UniProt KB is the most highly networked tool. It carries information, it gathers information from several sources, and it gives you best result, best possible information that you will be searching. So what is the site? UniProt. It is a this site is a combination of 3 databases: Swissprot, Tremble, and PIR. Protein Information Resource, very old database. Swissprot is a new protein database. And Tremble is European Molecular Biology Laboratory based database. What is the database? Collection of data. Who will give the data to the database? People like us. We do experiment, we deposit the data. So that is how all this data is integrated at one place with most possible information. Got it? So this is how you will get your search. So I have given the accession number, right? In UniProt, it will have a different accession numbers. This accession number I have got it from NCBI, right? National Center for Biotechnology. Because if you are studying in this college, your ID will be different. You will go to PG, your ID will change, right? Right? Or else you are studying here and you are doing some other course, some other place. So parallelly also your ID will change. Got it? Same way, same same organism, same protein, but ID changes in different databases. You should understand that part. Just click on the entry. Click on the entry and you just follow very thoroughly that it will give you best possible information. You come little front. It will give best possible information, that name of the protein and organism. This part we know very well. And GO annotation. What is GO annotation? Gene ontology annotation. Gene ontology. Ontology always, whenever you get this term, no, it is an indication of evolutionary relationship. Evolutionary relatedness. So evolutionary in the sense, not all the time with our forefathers. It is like comparison. This is actually new protein sequence deposited in the database. As such, there is no, as such, we have not written anything about this protein. Not many scientists worked on this protein. For example, say, still this database will give you a lot of information. How it will come? We know that all the proteins are made of hardly 20 amino acids. They are present in different permutations, combinations to give different proteins. So definitely, when there is 1 to 320 amino acids, particularly in this protein, some stretch of protein will be matching with some other protein of different organism. Some stretch will be matching with some other organism. Though we all are looking little different, but we are 99.9% similar. Got it? In everything. Same way, you will have this sequence matching with some other sequences. So this tool in the background, it is already compared and searched data, all readily available. But there are other tools how you can actually compare and try to understand. Those are BLAST and all that we are not taking up right now. But here, how you get this much information is by virtually searching all the available data based on its protein sequence, matching with some other organisms, proteins of some other organisms. Got it? Similar proteins. Clear? So, GO annotation, ontology, whenever you get to it, that whatever information you get there based on certain similarity searches, evolutionary relationships, like that. Okay. So, you can, when you have time, you just go through the names of these activity. This might have this activity, bolke, you will have several. It may have DNA binding, protein binding, and all, so many examples you will get. But anyway, if you come down little, so molecular function, most important. So molecular function, what it might be doing? Peptidase. What is a peptidase? Enzyme that degrades proteins is a peptidase. Clear? Any doubt? Then biological function. What this protein might be doing? Proteolysis. When we are trying to figure out some antigenic proteins, because we need to identify what are the antigenic toxic proteins from this organism, so that we can expect them, we can expect antibodies to be detected for that protein. So don't worry little about that. I will clarify your doubts. As of now, your aim is to identify most antigenic, most toxic protein from this organism. Identification of antigenic proteins from this organism, right? So if you identify most antigenic protein, what that protein might be doing? It will be doing harm in the host. You understand that part? So how these molecular interactions will happen? If yours is antigen, in host body, it will induce antibody synthesis automatically. It will. So how we can check presence of infection in the host? If you have antibodies to that specific antigen, it is like you got infected. So we don't know which protein of that organism is involved in producing antigenicity, producing immune response in that host. So this analysis will help you to figure out, might be these proteins are very important. By several analyses, you will be able to make out that they might be involved in inducing immunity in the host. You should choose such proteins, they induce immunity. They interact with your host. Then you will be successful in developing a kit and detection of an infected organism, infected host, whatever. Okay, so you read out all these things and you can make note of few things here. Then finally, but you will be able to make out very clearly that this is involved in proteolysis. In the host also, suppose think we are the host. An organism is infecting us. So what will happen? It will pinch here. Slowly it will damage. Slowly it will get entry and it will go interior. It will damage the organs. Same thing happens even with the Trichomonas fetus, where it infects genital tracts. Genital tracts. Slow, it will be first hiding. It will hide in the genital folds. It will not, if it is hiding, our body will not be able to identify, right? Little exterior foldings, it will hide for some time. What I understood from all this analysis is, and slowly it will secrete out proteases, especially cysteine proteases. It is hiding. It is not allowing entire protein parasite to enter into the host initially. Initially, it is secreting lot of proteases. So what happens? Those proteases will damage the lining of the genital tract. So little damaged condition, then the parasite enters. It secretes more proteins. Several of its membrane proteins altogether will damage the host lining. Uterine lining is damaged means very difficult to conceive. Even if it is conceived, it will abort. That is how the damage occurs. Got it? So this I'm able to explain based on whatever analysis I have done. So the similar analysis you will be simulating and we will come to them. Okay. So by using UniProt, what you are able to figure out very confidently that your cysteine protease 8, the first protein, is involved in doing proteolysis. Because our cells are made of large amount of proteins, right? So all the proteins will be damaged. Got it? Good. So still if you can, yeah, keywords regarding this hydrolase. Hydrolysis is also digestion, right? So hydrolase is proteases. And later on, if you still have time, you can access this site and you can little more study. Yeah, one more important thing. Because you know that proteins do not function in their primary sequence. The methionine and all that we write, no, that is a primary sequence. They will not do any function in the primary sequence. They will undergo secondary structures, alpha helices, beta sheets, and further they undergo to form a three-dimensional structure, then they will do the function. Got it? So what happens if they have to do the function in that elongated ribbon kind of structure? They may get damaged, right? First thing, they may get damaged. They may not be able to traverse through the organelles of the cell wherever it has to go. Very difficult, no? But something it is like three-dimensional structure. It can protect itself and it can also traverse several regions. You're getting? So this is a three-dimensional structure of the protein. Here you can little zoom. You can zoom and you can rotate and you can see. So what are these ribbons? This is alpha helical region. Sorry, just a second. I will show you. So this helical part is alpha helical region. Yes. So this is alpha helix. You have learned the alpha helix? And this particular protein has alpha helix as well as, and these vertical ribbons are beta sheets. Beta sheets. Beta sheets will be like sheet. Alpha helix is like helix. So this particular protein has certain domain of protein as alpha helix, certain domain of protein as beta sheet, and in between you can see several sequences. They do not have a proper orientation. They are called as connecting links. Turns and loops and turns. So this is a three-dimensional structure of your protein. So all this information, if I give you a project on cysteine protease 8, you can do within 1 hour. Because this is the authenticated site that will give you a lot of information about this protein. If you Google, will you get? So these are called as most authenticated and most informative data. So based on this, you will be able to write so much you can write. Here? Okay. Further, if you still scroll down, you will have which family it belongs to, family names, cathepsins are group of proteins, it belongs to that family, and several other related information. So currently, why we came to this database? We wanted to know the function. We wanted to know the function. So listen, there is a caution for this function. So what it says is we have not given anything. It is based on, if you read it, it is based on sequence similarity search and other organisms. But if you do some experiment on this protein in the lab, establish certain functions, so you can add all that. So based on, this is whatever related available data is based on purely predictions. But if you really do some experiment, you can add to the primary data. Got it? That is how database builds. Got it? So this is called as functional annotation. Same way we can also do for one more protein. Let me take example of a surface antigen like BSPA like protein. So you have to just take that. You should always have your accession numbers in hand, and you should always sequences in hand. So then all your job will be done. Okay. So surface antigen like BSPA like protein. Yes. So this also says this has taken all sequences from in a comparison one, because I tell you this is a recently sequenced organism. All this analysis, everything is possible, and our understanding also increases only by sequencing. Only by sequencing. Its sequence was found out recent 2017 like that. That is more recent when compared to other organisms. So just available as dump ATGC pura likha hua. Who will understand that? Slowly, people should start analyzing stretch by using several tools, and they should keep entering the data. Then only the information will develop regarding the whatever that is being sequenced. Slowly it is going on for this organism. So that is why you don't get primary information, but otherwise also based on several comparisons, you will get several information. So if you read out, you will be able to make out very clearly that. So you can see here very clearly, it has some catalytic activity, it binds to DNA, it is a virus receptor activity, it has an antioxidant activity, then it has a transferase activity, hydrolase activity, lyase activity. So such hydrolases, lyase activities are important for us. We are looking for a toxic protein, right? So we should check whether this protein has any, so it has a toxin activity. Toxin. So these are all catchy things you should note down from this, and you should write. Suppose you want to check whether this protein is involved in carbohydrate metabolism, for example. So if you are getting any such annotations, you should write down. Whether it is involved in lipid metabolism. You can see you have an annotation, it binds to lipids. So it is likewise, depending upon what you want to search. You can see one more here. Cell adhesion mediator. Is this property important for us? Cell adhesion mediator. So this, for example, this is your membrane, this is your BSPA protein, whatever region that you are mentioning exterior is involved in helping the parasite to adhere to the host tissue. Is it important? Because the parasite has to establish some, it should start there, no, in order to give the pathogenic lesions. So these proteins help in adhering the pathogen to the host tissue. So such things you should note down by looking at this table. So where from you're getting all this data? The database is comparing all the sequence and submitting. Comparing all the sequence and submitting to the databases and interconnected everything. Wherever we are searching, you will get this data. Got it? So finally, but anyway, if you come down, yeah. So by looking at this, you will be able to annotate, and it is also showing one more tab. Don't go to all these tabs. suppose if you are getting any biological function, molecular function, that you have to note down. Otherwise, you span all these sequences, all these sites. You just scroll this, read one after another, and you check any toxicity related, any host pathogen related, because we are looking at such things, right? Note down all those properties, right? So likewise, you should, and you see your UniProt also gives you subcellular compartment. Was this BSPA a membrane protein? Did I describe about that? So let us check anyway. So subcellular compartment, this particular protein is in the membrane. So this is what I explained just now. I will also explain you once again. Is that I will open the presentation and I will go to this part. This protein, right? So this is your BSPA protein. The faster sequence was given to TMMHM site, and it has analyzed all the sequence and it has predicted. So when you see the prediction, so these lines indicate the domains. So how many colors you have? You have a pink, and you have a red, and you have blue. 3 colors. So pink is indicating outside, red is indicating membrane, transmembrane, and blue is indicating outside. Blue is indicating inside. Okay. So when you look at this protein, large amount of protein is where? Outside. So you can see this table also here. By looking at this table, you will be able to make out that large amount of protein is here. And little amount of protein, 22 amino acids, passes through the membrane. And you have, this is exterior, and you have some membrane domain, and you have this blue one. Small amount of protein is present inside. How many times it is passing through the membrane? Only once. One vertical line, because only one vertical line you can see, no? That indicates one time it is going through the membrane. And the when you may also get some pseudo vertical lines. So you should consider that vertical line that comes up to the probability value of 1. I will show some more examples where you will not find vertical line up to the probability of 1 in this graph. You understand this graph now? So let me also tell you, because this part you should understand very clearly. This graph. You know that any cell, whatever your type, you know that parasite, whatever parasite you have a posterior glandular, you have anterior, you have posterior glandular, 3 anterior glands, right? And it has a membrane, right? Has a membrane. And now say in our analysis, you know that your membrane is a double layer membrane. Your membrane is a lipid bilayer, right? All eukaryotes. Particularly, this particular BSPA protein is transmembrane. So, as I would discuss earlier, suppose this is a double layer membrane, certain proteins may be, this is exterior and this is maybe outside and this is inside. The proteins may have only towards outside membrane, certain proteins may have only towards inside membrane. Certain proteins may travel through the membrane. So these are different possibilities, right? So your BSPA, whatever is there, it is showing how amount of protein present outside from 1 to some 3,500 amino acids. And some 22 amino acids transmembrane, small region is towards inside. So like these proteins are very important. Why this particular protein? If this protein is important for the cell, this large domain would have been inside, small domain would have been outside. But this is reverse. Our protein is towards outside, small amount of protein is towards inside. So what this protein might be doing? Definitely you can think of this has something to do here now. This protein is present here. This might be involved in interaction with the cell. Interaction with the cell. Got it? So it might be giving toxicity, it might be giving lysis, interaction, adhesion. So this protein might be helpful for parasite to adhere to the host cell lining by establishing communication. Clear it is. So this is how you should search for protein. At this point of time, when you want to search for some toxic proteins involved in pathogenicity, virulence, this is how you should search. Analyze a given set of protein and you should propose at the end of the project what might be the most suitable protein for our antibody detection technique. You're getting? Got it? Okay. So, let me also take you to TMMHM analysis one more time. TMHMM analysis. So, this predicts presence of transmembrane helices, right? So, this is the site and you have to paste your FASTA sequence. So, I told you, you have to keep your FASTA sequences ready all the time. Then only you can do, several times you have to do cross check and all that you have to keep on doing, right? So, you should keep all your FASTA sequences ready, then only you will be able to work out. Did I share FASTA sequences or you have searched out? Have you searched? Ready? Ready with you all the sequences? So, this is cysteine protease 8 and this is your, because in the search sometimes you may not get it as BSPA. How I got the name BSPA? Did you understand for this protein? I got this through when I did functional annotation in UniProt. Otherwise, in NCBI, it is showing it as hypothetical protein. Hypothetical means we don't know anything about this protein. Hypothetical. Got it? So, but how I could get the annotation is because I learned something. I understood something from UniProt. I extracted whatever information that was available in the UniProt. Got it? So here ends your sequence. Okay. For the time being, let us use only this much. Somehow I'm not able to copy that region. So this is your TMMHM. So you have to paste the FASTA. It is telling no, you should check the indicate what it is asking. Sometimes it will not ask FASTA. You have to give from your FASTA base onwards, and other times it will ask FASTA. So you should give as FASTA sequence. So what is the so how you can identify a FASTA sequence? You can identify based on, so most of the time you should make sure that you should not have any gaps in your sequence. If you want to remove any gaps in any words, whatever, just paste everything in the URL page. Then if you just copy, all the gaps will be removed. Okay. So better don't give gaps because the algorithms, they don't understand if there is gap means they will think there is no protein. So make sure there are no gaps. Okay. So then you have to submit. You need not have to do anything. You just do the submit. So it will take a moment and it will. So what this tool might be doing in this gap? It will compare your it will analyze all the sequence and it will give you the most probable result with, it is not loading, most probable result that what this protein might be doing, how much is outside and how much is inside, how many are the membranes. So it is taking a moment, but this was your result. It will give you this result. Here, this you will get. This is BSPA. So you will get all this information, the size, length of the protein and the number of transmembrane helices. TMMHMC's transmembrane helices is 1. Predicted, this is all predicted. Then you have how much is outside, how much is inside, how much is transmembrane, all this information you will be able to get. So did you understood how to analyze this? So let's go to the site and check whether it is done. Yes. Not this one. No? Still it is taking time. It needs fast internet. Okay. So this is how it will do the analysis, and we'll go back and we'll redo the submit once again. Yes. Somehow it is escaping. It is loading, but it is not, somehow it is missing. Okay. We will practice once again. So you will get that output and you should know how to analyze that output. So this finishes your TMMHM analysis. Right? You understood TMMHM analysis. And next, now you understood your protein has some function related to antigenicity, virulence, cell adhesions, whatever. And when you understand it, but these are very big proteins. You may not use the entire protein for developing, synthesizing antigen sometimes. So you should choose what regions from that protein might be antigenic. Because I showed you three-dimensional structure, right? Certain sequence of the protein will be inside, in inner folding. Certain sequence of the protein will be towards exterior of the protein. Within the protein also, within the 3500, whatever is there, certain regions will be hiding inside. They might be doing some other function, binding a metal ion, many things. Outside, what is exterior most of the time will be involved in, that will be exposed out automatically. That will be involved in either antigenicity, adhesion, whatever. So there are tools available for you using which you can predict your sequence. You can predict most antigenic sequences, because now at this particular point we have to identify some proteins that are antigenic. They should be antigenic. So we use some resources called as IEDB tools. What are the tools? IEDB resource analysis. So IEDB resource analysis. So IEDB is nothing but immune epitope database. What is IEDB? Immune epitope database. So what are epitopes? Regions of the proteins that induce immune response in the host are called as epitopes. Epitopes induce antibody response. So within the protein, epitope regions are small stretches of that particular protein actually induce antibodies. Epitope binds to antibody. That part you should understand. So in this bulky protein, we should identify what are the epitopes. Epitopes are nothing but small stretches in the protein. They will be actually involved in immunity. What is antibody stimulus or binding to antibody? Both ways. If you provide, it will bind. If not, it will stimulate. Both ways it is. So here you have, we learned about immune cells, right? T cells, B cells, and we learned about cells involved in innate immunity. Neutrophils, eosinophils, all those are involved in innate immunity. Lymphocytes are involved in acquired immunity. In acquired immunity, you have B cells and T cells. B cells will produce antibodies. T cells will be cytotoxic. They kill the pathogenic cell, whatever. So now, as I mentioned that we have to look for, means our antigen should bind to B cells. Because B cells only will produce antibodies. You're getting the logic here. So which kind of epitopes we have to predict in our proteins that bind to B cells, not to T cells, because we want to identify antibodies. So I will clarify this later. And we have to predict B cell epitopes. B cell epitopes. In our protein, which regions of your protein will interact with the B cells? So what B cells have in turn? In turn, B cells will have antibodies. and some regions of your protein will interact with the antibody and will induce its resistance. Got it? So we have to identify in our protein what are the different regions that bind to the B cell or antibody. Directly, it is antibody that are produced by the B cell. Right? So in the, you have several tools again, depending upon what you have in your hand regarding the protein, whether you have a linear sequence, whether you have the structure, not predicted structure, solved structure. But confidently what we have is our protein sequence. Based on what we have in our hand, we should use the tool. So what we have in our hand regarding this protein, we have sequence in the FASTA format. So we should paste the sequence. So sometimes this also will not mention, but you check by giving FASTA only. If it is not working, again you should take from first amino acid. So choose the first algorithm, biprid linear epitope prediction to 2.0 version. This is older version and this is new version. What is the name of the tool? Biprid linear epitope prediction. Got it? Just give submit. So it is not accepting invalid characters. So what you should do, remove all this. Okay. So don't remove any of your amino acids. Should be very careful. But blind thing is first amino acid always will be methionine. Okay. So you can remove all the remaining things, greater than symbol, accession number, name of the organism, all that you can remove. So since this is a very big protein, it will take some time. Meanwhile, we will also do for one more protein. One more small protein. We will do for cysteine protease 8. This is a very big protein. It will take a little moment. Okay. What happened? It is searching, right? It is not allowing us to do another one. So we have several sequences in our hand, right? So we have our cysteine protease, this one. So let's copy this. So sometimes you may have gaps. So what you should do is paste it at the URL. All the gaps will be removed. And you give your sequence. This is a very small sequence, so it may not take much time. So both are running parallelly. Internet is relatively slow. So it will take a moment. It will take a moment and you will get some graphics, you will get some tables. How you got for TMMHM, here also you will get. And meanwhile, we will go to the presentation. Yes. So likewise, for all the 10 proteins, you have to do TMMHM analysis and you have to get the graphs and you have to make a table. So here you can see a table. Here you can see a table. Okay, the table is missing. I will show you the table. You have to mention, yeah, this is the table, not this table. The table is missing. Okay. So you have to make a table like this, showing how much is the exterior, how much is the interior, how many membranes, all that. There is one table that you have to make. And we will go to IED. Yeah. This is what you have submitted right now, right? And you will get the graphics. You will get the graphics. So there will be a threshold 0.5, default threshold. So regions that are falling above 0.5. Take this example. So you will get this graphics above the regions that are falling 0.5 above. And here you see amino acid sequence is starting from 1 to 320. So in the 320 amino acids up to almost say up to 10, for example, it is below the 0.5 threshold. They are not antigenic. So here you can see some region up to 40. Yellow will be antigenic. So likewise, this is some non-antigenic region. This is a very big antigenic region. From here, for example, from 70 to 120, like that. And here, this is not antigenic. This is antigenic, like that. And table. So just by looking at here, you cannot figure out correctly which amino acid is antigenic, which amino acid is not antigenic. This itself will generate a table. This to this is the most predicted antigenic site. This is the length, likewise. And you should figure out all this, and you should choose the most lengthiest epitope domain. These are called as epitopes. An epitope can be as small as 6 amino acids. 6 to 20 is called as an epitope, always. So you should choose largest one. Largest one, and means all these are important, but you should mention it has a largest epitopic region of predicted epitopic region of 53 amino acids. Clear with this? So this is how we have to analyze for each protein, and we should figure out antigenic domains of, yeah, here are the graphics. So what is the sequence we have submitted here? Cysteine protease 8. 320 amino acids. Can you see 320 amino acids are analyzed? And you can see the graphics here. So here it will tell you some score. Epitope. This might be epitope, this might be epitope, like that. There will be one more elongated table and it will give some score. You don't need to bother about this. So all the scores, whatever epitopic regions will have more than 0.5. You are able to see? So this is more than 0.5, like that. So this you don't, all this is consolidated here. Okay, so likewise, whatever epitopic regions will be predicted and you will have a very smaller table. Got it? So this is how you have to analyze and write down the epitopic domains. So let's see whether this protein is also analyzed. It is a big protein, needs little fast internet. So it will do. So I'll show you the BSP ECA. Very big, and you will not be able to make out anything. You will not be able to make out anything from the graphics because it's a very large protein, right? 1 to 3,500 will have to be present side by side. So in a short space, this is little complicated diagram. Okay, but anyway, you will have the table now. You will have the tables. Like this table will be there. From the table, you can figure out what are the different largest domains that are present. Got it? So this is how you will be doing the epitopic domain analysis. And finally, yeah. So, but from this study, though I analyzed 10 proteins, I did functional annotation, I did cell membrane localization, properties, epitope, GEVO annotation, then epitope analysis, all that I have done. But I studied little papers, background papers of this organism. Found out that this particular organism is not entering into the host tissue directly. It is secreting lot of cysteine proteases. Conclusions. Lot of cysteine proteases it is secreting and causing toxic damage within the host tissue. Then it is taking entry and involved in all the pathogenic lesions what we studied. So for the initial study, I have used I found out cysteine protease is the most suitable protein. When pathogen itself is not entering into the host initially, if you choose a BSPA like protein, in the later stages, they might be useful as a toxic antigens for you to detect antibodies. But you will miss out initial detection if you choose only BSPA kind of proteins or any other membrane proteins. But if you choose cysteine protease 8, you can detect presence of the infection in the early stages. Detection of early stage is very important. Because after causing damage, if you detect, what is the use? There is no use, right? So any infection need to be detected in early stages. So if you choose cysteine protease 8 based antigenic detection kit, you will be able to detect cysteine protease 8 induced antibodies in the host even from the early stages of infection. If you identify that particular host infected, you can eliminate that host, that cattle from the herd, and you can separate it from the herd. You can prevent further transmission of the infection in the herd. That is our aim, right? Because what is happening, one infected animal in the herd can cause 20 to 90% of the infection. If all the animals are infected, you will have very low fertility, very low yield from that herd. So what is the use of a farmer maintaining that much, so much money, time and all that, right? Resources. So early stage detection is most important, and cysteine protease might be most suitable for the beginning. So, and if you study the literature, multiple types of cysteine proteases were described, multiple, almost like 22 like that, by several scientists and their papers. And these parasites tend to inhabit the genital foldings in the early stages of infection and secrete proteases, and they get invasion. Got it? And transcriptomics, proteomics studies. So you understand what is transcription, mRNA synthesis. Protein is protein synthesis. Analysis of mRNA and protein of this organism also highlighted presence of several cysteine protease related mRNA, cysteine protease related proteins identified in the analysis. Several high copy number was identified. So hence, and among several multiple cysteine proteases, especially cysteine protease 8 is highly expressed. These are all several cysteine proteases that were identified to be present in this organism. Cysteine protease 8 is the highest, followed by cysteine protease 16, cysteine protease 13 and 16. 13 and 16, right? So these three are the most among all 20 cysteine proteases. So all the cysteine proteases will be involved in damage, but which is major cysteine protease involved in causing more damage? So how you can identify if they are present in large number, they will attack more. So that is how by transcriptomics, proteomics studies will help in understanding the levels. We call them as levels of number of mRNA. You have many. Definitely you will get lot of proteins. If you have many many proteins, anyway they will do the function. So total mRNA and total proteins will be involved in giving you more analysis and identification of the suitable thing. Okay. So this will be your conclusion, and yeah. So we will stop it here.